Okay, so from our initial 6 fragments, what we were able to do was to rapidly sequence the complete genome of a new polyomavirus that we named after Washington University. We call it WU polyomavirus. And shown here is the genomic organization of a canonical polyomavirus. We found on the left side here is what's called the early region. It encodes a large T antigen and a small T antigen. These are formed by differential splicing. And on the right side is the late region, because it's transcribed late in the life cycle, and these encode the viral capsid proteins that are important for assembly of the virus, the virions themselves. Okay. So the question we wanted to know was, well, really, we have this idea from the blast that it's novel. Well, let's look phylogenetically to define exactly how novel is this. And so what you see here on the left side is the large T antigen, which is a protein that's highly conserved and required for replication of known polyomaviruses. And, okay, I'm going to be bold and try this again. Color. Touch the color. Touch the color. Touch the color. There you go. Okay. And so here, this is known as the primate clade of polyomaviruses. And what you can see is that WU clusters closely to that in the large T region. So here's the VP1, the capsid region, and what you can see here is that the length of this branch is now much larger, and so there's something going on here that WU seems to be much more highly divergent, relatively speaking, in its late region compared to the early region, relative to all the other polyomaviruses. And so this was sort of curious, and I'm going to come back to that in a second. So this was all very exciting for us, and what concerned us was that just as we were about to submit this paper for publication, was that there was a report that was published by a Swedish group led by Tobias Allender who did a very similar approach. They used high-throughput sequencing on pooled respiratory samples, and they claimed to have found a novel polyomavirus that they found in respiratory secretions of 6 patients. Now, you recall, BK and JC are the 2 known human polyomaviruses, and they were both described in 1971. And in the 36 intervening years, no one had ever reported another human polyomavirus. And so we were quite worried that we had actually just gotten scooped. But lo and behold, it turned out their virus, which is called KI, is shown here and here, is a sibling virus of WU, that they share about 70% amino acid identity to each other, which is less similarity to each other than BK and JC have to each other. So BK and JC are more similar to each other than WU and KI are to each other. And you can see here that they clearly still clade with the so-called primate group of polyomaviruses. And then out here in the VP1 region, what you see is that KI, like WU, has this very distant relationship. And so in a way, they corroborate each other in that we were worried at first that the first distance was some kind of alignment artifact or something like that. But this tells us that we think fundamentally there's something different about the biology of these capsid proteins, that they've been subject to some other kinds of selective pressure or evolutionary process. Okay. So, again, we found the virus originally in one patient, this 3-year-old child with pneumonia. The second question we wanted to ask now was, well, can we find this in other patients? And if so, how frequently? And where else can we find it? So we looked in respiratory secretions, and in our initial study, we looked at samples of acute respiratory illness swabs that our collaborators in Australia had, and we found about 3% were positive. We also looked in a cohort in St. Louis, and one was a pediatric cohort. We found 5 positives. The second one was a primarily adult cohort, and we found only 1 positive. And so then this led us to bias our search in the future to more pediatric populations. And so we did a second cohort in St. Louis that spanned a 12-month period of time that was focused primarily on pediatric cases. And we found almost 3% positivity by PCR in this cohort as well. So what we know now from our studies and from a number of other studies is that this virus is globally widespread. They're all sick. So there have been reports now from independent labs from countries all around the world that show that, again, looking primarily in respiratory secretions from patients with respiratory illness and predominantly in pediatric populations, that WU is present in a fraction of those. And similarly, KI virus is also present. Okay. And so here, just looking at some of the age distributions and some of the interesting observations, so we found that the highest rate of infection that we observed was in the age group between 7 and 24 months. We did find in our youngest age group, we did find that the lowest rate of infection was in the 0 to 6 month age group, which suggested to us the possibility that there may be at least some protective maternal antibody, but that this was clearly not complete because we were getting positives. And in fact, we had a 1-day-old infant who was delivered by cesarean section and immediately intubated for respiratory problems, who their initial sample was positive for WU, and so our suggestion or inference from that is that presumably there's maternal transmission in utero, although this is again just a case report. At the other end of the extreme, we did have our oldest patient was 53 years old, and again, more anecdotally than anything else, we had 9 patients who were positive for WU in our various cohorts who were older than age 12. And in 8 of those 9 cases, there were clear clinical rationale or documentation of immunosuppression. A couple of these were HIV patients, a couple were transplant patients, et cetera. And so this is, again, more anecdotal than anything, but it establishes a model where perhaps, like the other polyomaviruses, you have early infection in life, you may have persistent infection over the course of your lifetime with periodic reactivations. This may be more frequent in immunocompromised populations. And so that's our sort of working model right now. And so here's another case report. And so in our series of about 2,000 respiratory samples, we had multiple samples collected from a couple of individual patients over a 4 to 6 month period. And here is one patient. This is a 16-month-old liver transplant patient. And this patient had a total of 5 nasal swabs collected over the course of this period of time. And what we found was that during the hospitalization period after transplant, that there were 3 samples that were continually positive for WU virus using a real-time PCR assay. And so, one possibility was, of course, that, well, the patient's immunocompromised. Maybe they just can't clear the virus very well because of their compromised immune status. But I would note that we did also test for other respiratory viruses, and so at different points in time, they had other bona fide respiratory pathogens like adenovirus and rhinovirus, and they're perfectly able to clear those because they are not observed following at the follow-up visits. So they're not completely incapable of mounting an immune response. Do they have respiratory symptoms each of those time points? Or? They do, yeah. So they did not have respiratory symptoms continuously throughout, though. So they had symptoms, they waned, they had symptoms, and they waned. Okay. And so another possibility we thought about was, well, maybe it's just slow to clear, and maybe there's just this gradual decrease, but we're using real time, and so maybe it's very sensitive. But it turns out that there's actually constant copy number based at least on CT values throughout this middle 3 time points, so suggesting that the virus is still replenishing itself or is at some equilibrium with the immune system. And that it is capable of undergoing some type of ongoing replication. Okay. But eventually, they do clear. And so at this time point, there was no evidence for WU polyomavirus. Okay. So I told you at the beginning that the hallmark of BK and JC virus is that you can detect them in between 5 and 20% of the normal population in their urine because of the reactivation. And our first assumption was that, well, we should look in urine and we're going to find lots of WU polyomavirus. So we looked by PCR at about 200 urine samples from Australia, and we didn't find any positives. We looked in St. Louis at 500 urine samples, and we didn't find any positives. And so we don't know what to make of this, other than our first hypothesis here is that, well, there's something fundamentally different about the tropism of these viruses from BK and JC virus, which are very easily detected in urine. And of course, this may not be the appropriate cohort, but these were actually, in our St. Louis cohort, a population of renal transplant patients who have been followed, and we know we were able to detect BK in a fraction of these, and so we know about the integrity of the samples and so forth. Okay? But again, even detection of this virus in respiratory secretions is not part of the paradigm with human polyomaviruses, that BK and JC virus are not frequently described in respiratory secretions. So we know already that the biology in some way is just fundamentally distinct.